So in this video we'll be mainly covering the antifungal agents and mainly we'll be going through its mechanism of action only. Since it's the first video, I'll be only going through the mechanism of action. Soon I'll probably make another video which covers different aspects such as its adverse effects, dosing, etc. So in this video we'll be just going through the mechanism of action to understand how these drugs actually work in our body. So as you can see, the antifungal agents are divided into 4 classifications, which are antibiotics, antimetabolites, azoles, and allylamine. You have to note that these classifications also contains sub-classifications, which I have not written here, but I will explain once I go through those topics. So coming to the first one, which is antibiotic antifungal. This is the antibiotic antifungal. So to come to the first one, you can see here. You can see here that the antibiotic antifungals are divided into 3 subclassifications, which are polyenes, echinocandins, and heterocyclic benzofurans. These 3 are the antibiotics. Now you all must be wondering, why am I teaching each one of them separately? I can just explain it as, antibiotics as a whole. The reason is that each subclassification works in a different mechanism of action. They all have different ways of acting in the body. So therefore, I'll be explaining each of them separately. Coming to the first one, which is polyene antibiotics. As you can see here, there's a structure of polyene antibiotic. It contains a lipophilic side and a hydrophilic side. Now, to come to the important part, that is where does it act and how does it act? Well, it acts on the fungal cell. To be more precise, it acts on the cell wall and the cell membrane of the fungal cell. So as you can see here, this is the structure of the cell wall and the cell membrane of a fungal cell. Now you can see 3 words over here, which is beta-glucan, chitin, and ergosterol. Now for those who are wondering what these are, beta-glucan and chitin are basically a polysaccharides found in the cell wall. Now what do they do? Now what happens is these 2 structures, they form a cross-link. Now what is a cross-link? You can see here that they are interacting with each other. So that basically from this cross-link, what happens is it provides a sort of rigidity to the cell wall, making it more strong and structured. Now ergosterol, if you're asking what is ergosterol is nothing but a steroid molecule. It is a steroid molecule found in the cell membrane. Now what is it crucial for? It is crucial for maintaining what? It's crucial for maintaining the structure, crucial for maintaining the fluidity, and it's crucial for maintaining the functionality in fungal cell, obviously. So as you've understood this, coming to the point, now you see this white molecule over here. Don't worry, it's nothing but the polyene antibiotic. That is the drug you take, polyenes. Now what happens when you take it? Now, so basically, as you can see here, it goes into the cell membrane, and what happens after it enters the cell membrane? Well, that is quite interesting. Once it goes to cell membrane, as I told you, this ergosterol component, it binds. The polyene antibiotics bind to this ergosterol. As you can see, it binds. So I'll draw one more over here. It binds to this ergosterol, and what happens? Well, it binds to the ergosterol and it forms, it joins and it joins and it joins. Might have to wait a minute for this. It joins, and you can see it forms a pore. That is a leakage in the cell membrane. Now, what happens when a leakage occurs in a cell membrane? Eventually, yes, the fungal cell loses a lot of its, major stuff from the cell to outside. Yeah. So that's what happens. So what do you call this term? This little hole over here. Well, you call that as a micropore. This is a micropore. So this drug binds to the ergosterol and it forms a micropore. And after that, what happens? After that, as you can see, I've already drawn 2 arrows over here. Now you must be wondering you must have already understood what happened. So I'll just draw the micropore again. You can fast forward if this is boring. This part of drawing. Now once this micropore is formed, the fungal cell loses a lot of important stuff, such as what? Such as important ions. It loses a lot of ions, it loses a lot of amino acids, and it loses a lot of other stuff. Now, if you're wondering what this other stuff is, it can be anything which is vital for the fungal cell, such as cytoplasmic components. So what happens after it loses all this? So I'll just draw this. I'm doing this because I don't want to write again. It loses all this stuff outside the cell. So what happens once the fungal cell loses all this? Well, once the fungal cell loses all this, it ultimately leads to a state where the cell, it affects the cell's membrane. It destabilizes the membrane potential. And what happens once this occurs? It ultimately leads to nothing but cell death or cell lysis. So that's how basically this first subclass of polyene antibiotics work. As you can see, it's the polyene antibiotics. Now we are coming to the second one, which is nothing but echinocandins. Ta-da! So as you can see, this is the echinocandins. Now this is the second subclassification of the antibiotics. Now it is comparatively, as I've written here, it's comparatively a new class of semi-synthetic drug, and this is what echinocandins look like. And it's not exactly how it looked like, but it's how I'm going to depict in this video. So wherever you see those dots or the structures, remember it's the drug. So yeah, so again we are coming to the same structure. Why am I showing the same structure as before? Because this drug too behaves in a similar way, not exactly same, but it does behave in a similar way compared to the polyene antibiotics. So you have seen beta-glucan, chitin, ergosterol, and you have seen a new compound, which is beta-glucan synthase enzyme complex, or in simple words, beta-glucan complex. You can say that. So what does it do? What is this stuff? What is it actually? Well, I'll explain right now. Well, this beta-glucan synthase complex is nothing but a complex. It's there in the name itself. It's a complex that is, to be more specific, it's an enzyme complex. It is an enzyme complex for what? It's an enzyme complex responsible for synthesizing synthesizing beta glucan. I know it's pretty simple. It is simple. I mean, it's there in the name itself. So this structure is what produces the beta glucan. Now this is the beta glucan. So this purple thing produces the beta glucan, which travels to the cell wall, and as I told you just few minutes before, is responsible for the cell membrane integrity and cell wall integrity and etc. So coming to the mechanism of action. Oh yeah, this is nothing but same stuff which I have written. This is the beta glucan. This is beta glucan complex. I'm just repeating what I've told just now. Produces what? What is this? What does it produce? It produces what? It produces beta glucan. And what does beta glucan do? It binds with, what is this stuff? As I told you before, it's nothing but the polysaccharide chitin. What does their cross-linking lead to? Their cross-linking produces what? Rigid structure. Now, coming to the mechanism of action, it's very simple. I'm not gonna take much of your time here. Now you take this. This is what is this? It is Echinocandins. Echinocandins. See, it's Echinocandins. And imagine you take this. You have fungal infection, and you are advised to take this. Once you take it in, it enters the cell. Enters cell. So I might need a minute to actually take out this color. I don't think I have exactly this color. Oh yes, I have. So you take this drug, and what happens is, so you take out this drug, and it goes into your cell membrane, and it binds to this complex over here. So as you can see, it binds to the complex. And it's not that hard. I mean, what happens next is pretty much obvious to you all. So it comes over here, it binds. Actually, I was supposed to draw it in this step, not the previous one. So I'll draw it again. It binds over here, and what happens? Ultimately, it leads to the inhibition. That is, it inhibits this complex. See, it inhibits the complex. Throw an arrow to show the next step. Yeah, so it inhibits this complex, and therefore what happens? So in this new step, you see the difference. You don't see the blue strands over here. You see the blue strands over here. You don't see the blue strands over here. Why? Because the blue strands, which is chitin, is produced. The blue strands, which is beta glucan, is produced by this beta glucan complex. So since the drug which we just took in, which belongs to the class of echinocandin, blocks this complex, the complex can no longer produce the beta glucan. Therefore, the cell wall, it's kind of empty. Not empty as such. It does have chitin, yes, but there is no beta glucan. Now we must be wondering what will happen. It's not a big deal, Well, it is a big deal. Like I said before, you need both. You need chitin as well as beta glucan for the cell wall's rigidity. You need both of them to make the cell wall strong. So without the beta glucan, there is no beta glucan. What happens is, without the beta glucan, laws of cell wall rigidity and leads to a lot of factors and ultimately causes cell death. The reason I'm rushing is because I've reached the end of the space. I have come to the maximum space I need. So this is basically how it works. In simple words, it inhibits this stuff which is producing the beta glucan. Therefore, there is no beta glucan over here. As you can see, no beta glucan. We need beta glucan and chitin to form cross-linking, which provides rigidity and strength to what? To the cell wall. Now, without that, the cell wall becomes weak. It becomes weak, and it slowly it undergoes a lot of stages and ultimately leads to the cell death. Now coming to the next one, which is heterocyclic benzofurans. Just minimize it because I think it's a little bit big. Okay, so you can see the next one, which is heterocyclic benzofuran. I've written a few points for your reference. It's extracted from Penicillium, Griseofulvin. It has an advantage in where its resistance is observed very less. It has rare occurrence of resistance. Now, it's not, these informations are not important to understand how it works, but it's just some additional information which I've given. Now to come to the heterocyclic benzofuran, one thing I would like to say before I come to the mechanism of action is that compared to the previous two subclassification, heterocyclic benzofurans work in a completely different manner. They do not work on the cell wall or the cell membrane. Rather, how do they work? Good question. I'll come to it right now. Coming to the mechanism of action, this is the basic structure of a cell wall. You can see cell wall, cell membrane, nucleus, endoplasmic reticulum, mitochondria. This is a fungal cell, by the way. Golgi apparatus, lysosome, and you can see these structures are not actually important for us. The one structure that is important is this one. You see this small structure over here? Yeah, that is known as microtubules. And by the way, this is Griseofulvin. I just simply drew a triangle to show. I need not represent Griseofulvin the exact same way in the following video, but yeah, this is Griseofulvin. So it acts just in this structure called microtubule. So you just need to remember this one thing. This is the important part. So I'll just enlarge the microtubule. You see, this is microtubule. Does a lot of where that keeps popping up in between. So as you can see, this is an enlarged version of the microtubule. You can see a lot of small structures over here. Well, these small spheres are called as tubulin. These tubulins are the building blocks of microtubules. Now what happens is, this is Griseofulvin, by the way. You see this drug? You see something of, what? Oh, good. I'll just put it over here. And I'll put this over here. Wait, my bad. I'll just duplicate. So I got one more, and tada, magic. So yeah. So what happens is Griseofulvin comes and it binds to this tubulin structures of what? What are the tubulin structures? What are they part of? They're part of the microtubule. I'm just writing again so that you don't forget. Microtubule. Now I know I'm going a bit slow. This is my first video, but I'll try to catch up soon. So this is the microtubule, and the Griseofulvin comes and binds to the microtubule. What happens next? Wow, there's plenty of... Okay. 1 second. Yeah. So once it binds to the microtubule, I would like to take on another story, because I just want to explain how it works. So you understood. If you understood till the part over here, you have to go through the next phase, which is the cell division. Cell division, or another word, which is mitosis. Now you must be wondering, the drug came and the drug, binded to the microtubule. Now what? How does it actually cause the fungal cell death, or how does it act, or what is its significance? Now to answer all these questions, I have to explain the cell cycle over here. So as you know, as a revision, there are basically, you have the first one, which is known as prophase, involved in the cell division. And in prophase, what happens is, you have the nucleus over here, is genetic material, basically scribble something over here, You have two structures over here. Now, I know I have to use different colors, but I think to save time, I'll just go with white. Now you have, structures like this. So this is basically what prophase looks like. Now I'll explain the important parts, which is, you see these two structures over here. This is known as, I'll write in capital because it's important, centrosomes. See, as you've seen here, centrosome. Now if you're asking me what is centrosome, I shall explain. Now centrosome is nothing but, centrosome is basically an organelle which is found in animals that plays a role in organizing microtubules during cell division. So to put in simple words, they help organize microtubule during cell division. They help organize microtubule I've written microtubule, actually no, it's not microtubule during cell division. This is the most simplified definition. So they are just stuff which organize the microtubule. Now if you're asking me what does microtubule do in a cell division, I'm coming to the point. Now we have the next phase. What happens is, now this is the next phase. Since I was writing on the left side, I'll just write the stage over here. The next phase after prophase is known as prometaphase. Prometaphase. This is prometaphase. Now you must be wondering what happens in this step. Now in this step, what happens? You see this centrosome which I've mentioned before. Centrosome is there. Yeah. Now this centrosome, in the middle you have, I know my drawing is not that great. So what happens over here in this step is that, as you can see, in this step, the centrosome will reach out to these structures using what? Using nothing but, now these structures which is pulling the genetic material is, just make it more darker because it's important. Now these structures which you can see here is known as what? Known as microtubule. You must be wondering why it took so long to come. So it's not there in this step, but in this step, yes, microtubule is there. Now what is it doing? Microtubule is basically pulling this genetic material to each half of it. It's pulling the half of, so this microtubule is pulling half of this to the side, and this half microtubule is pulling half of this to the side. This is how it works. So if you're asking me where is the drug acting? Well, the drug does not exactly act here or here. I mean, prometaphase, yes, there is a chance, but the cell will still go to the next stage. Even if you take the medicine, suppose you take this drug and this is the fungal inside your body, it will still go to the next phase. It doesn't stop here. Doesn't mean that the fungal will succeed or the drug will fail. No, we are only reaching to the point where the drug will act. And this, my dear friends, this is where the drug acts. The drug acts in the metaphase. So remember, you take the drug acts in the metaphase. Now what happens in the metaphase? Metaphase is actually similar to prometaphase. The only difference is that in prometaphase, the microtubule binds to the genetic material. It binds, or I will say, it initiates to attach to this metaphase. Now in in the metaphase, what happens is the same structure actually, just it becomes more structured and see, in metaphase you actually undergo the process where it kind of separates the genetic material. This is what happens in metaphase. Prometaphase and metaphase are kind of similar to each other. They do have the slight variation. So the drug mainly, it causes its effect in the metaphase, which means that the drug will not go further. See, you see this phase? This is nothing but this is the next phase, which is telophase. So basically what happens is over here you get one half of the chromosome, over here you get one half, same here goes. So it pulls away, the centrosome pulls the chromosome, the centrosome actually pulls away this genetic material into two halves. And the cell, you can see from the structure itself, it's slowly preparing itself to divide, It's slowly preparing itself to divide and it pulls. This is the end product of metaphase. Metaphase, it attaches and it initiates the pulling process. And this phase is known as telophase. Anaphase, my bad. This is anaphase. Anaphase. Now, this is not actually important. You go on anaphase and this is called as telophase, where the cell completely divides. It's telophase, not telophase. And what happens over here is that it completely divides. You get a genetic material. You get a genetic material over here as well, and you have this chromosomes. So this is telophase where the cell completely divides, and you have the anaphase where they pull it apart. But I'll tell you one thing: anaphase and telophase are not important in this. Because why? Because the cell division will not go beyond metaphase. Because, like I said, the structure which binds to the genetic material over here, you see the microtubule. This is microtubule. Now, if you go back to the beginning of this, not the beginning of the video, but the beginning of the subclassification, you see the stuff over here, which is a part of this. See, microtubule. So what happens when you take the drug? It binds to the microtubule and it inhibits the microtubule. So what happens is the fungal cell is still active. Fungal cell is like, let's undergo the cell division, and it starts cell dividing with the first phase, prophase. And by the way, the structure over here, this long lines which I told you, it's known as it's got mistaken. It's not that. No, I was going to explain about the other one. So coming to the prophase, this is the first one, which is the prophase, and then we have the fungal cell. The fungal cell is starts to fungal cell is like, let's divide, and the fungal cell, nothing happens. When you take the drug, nothing happens. The fungal cell is like, let's divide, and the fungal cell goes to like prophase, and prophase, what happens, it goes to the next stage, like nothing happens. This fungal cell is happy, it goes to the next stage. And by the way, this is nothing but 1 second. And this is, I forgot to mention, this is nothing but chromosome. This is chromosome. And the fungal cell is like, let's divide, and fungal cell binds to this. It goes to prometaphase, and it still goes to the next phase, by the way. It goes to metaphase, where it binds and it organizes. It's ready to pull. But that's where the entire process goes wrong. Because the component which pulls the chromosome apart is microtubule. And the drug is already the Griseofulvin. Let's take an example of a drug which belongs to this class, Griseofulvin. Griseofulvin already has binded to the microtubules and inhibited. Therefore, this stuff or the structure, the microtubule, will not work since it's inhibited. Therefore, the chromosomes will not be pulled apart, which leads to what? Which leads to a stage called metaphase arrest. So as the word suggests, the cell division will stop right at metaphase. It will not undergo division. It will not go to anaphase where it pulls apart, and it will not lead to the ultimate cell division, which is telophase. It will ultimately, cause a failure of the cell division. And once, a failure of the cell division causes, it can no longer divide. So what happens? Fungal cannot divide. So basically, if they do not divide, they eventually die. So actually, I would just eventually fungal cell. Eventually, I'll write it as fungal. Eventually, cell death. Okay. Now, just put it in the center. Now, this is just one of the way how it works. This is just one of the mechanism. Now, there is an additional mechanism. Now, the additional mechanism of this class of drug is that it basically what it does is nothing but it binds to keratin. It binds to keratin. Then what? It protects the newly formed keratin. Not the old one, the newly formed one. You must be wondering, how does it protect the newly formed one? Well, it protects the newly formed one by binding to the keratin's precursor. I hope my spelling is correct. I'm pretty sure it's wrong. Yeah. Well, it binds to the keratin precursor, and therefore it protects the newly formed keratin cells. So this is just an additional mechanism of action of this class. It has two mechanisms of action. One is that it acts through this way, and second one, as I mentioned, it interferes with the fungal cell division and causes the cell death. Now, I know I've taken a lot of your time. Now I'll just hurry up with the next one, which is antimetabolite. We have come to the second classification, and this is an example of the drug belonging to the antimetabolite classification. This is an example, and the name of the example drug is Flucytosine. So we'll be using this as an example. So what happens in Flucytosine? This is the fungal cell membrane. So Flucytosine enters into the fungal cell membrane. Now, it doesn't just simply enter into the fungal cell. It enters with the help of something called as cytosine deaminase. Now, it uses the help of a molecule called cytosine deaminase. Now, for those who are wondering what this is, it is nothing but a transporter. It is nothing but a transporter molecule. Now, what happens is that this is found in the membrane as well. It's a transporter molecule which is found in the membrane. So what happens when this enters into the cell? Well, this is where it gets quite interesting. Now, the flucytosine has entered, okay? So this is flucytosine. So once flucytosine has entered, initially it undergoes metabolism. It gets converted. I'll just put a box. It gets converted or metabolized into a substance called as 5-fluorouracil. 5-fluorouracil, or to make it abbreviated, you can use it 5-FU. Now, flucytosine is converted into this metabolite by what? By an enzyme known as cytosine deaminase. Cytosine deaminase. Deaminase, resin deaminase. So after the, so once it gets converted into 5-fluorouracil, the drug or this metabolite has two options. It has two options to which it can follow. Now one option, which I'll depict as A, will lead to RNA synthesis inhibition. And if it chooses the second path, it leads to DNA synthesis inhibition. Now, to elaborate this mechanism of action, this is the two pathways which this metabolite has to choose. Two path, can choose any one of them, but key thing is that any one of them or both of them will ultimately lead to the lysis of the fungal cell. So let me explain the first one. So what happens in the first path? So imagine the 5-fluorouracil picks this path. So what happens is the 5-fluorouracil gets converted into 5-fluorouridine triphosphate. Fluorouridine triphosphate, or in simple words, I'll have to give it as 5-FUMP. 5-FUMP. This is an abbreviated version. So it's just a cycle, you have to remember. Now what does it do? Now imagine this is 5-fluorouracil is this spherical structures. Okay, so what happens now? Well, now the fungal cell, so this is the fungus, the fungal cell, fungal cell. So fungal cell is happy because what happens? Because fungal cell thinks or it mistakens, and it mistakens this structure for what? So this fungal cell basically mistakenly incorporates this 5-fluorouridine triphosphate into the RNA. So it thinks, yeah, uracil is back. So it basically mistakens these particles for uracil. So fungus thinks that this 5-FUMP is uracil and it takes it inside. So once this stuff is, that is the 5-FUMP is inside the RNA, so it takes, what happens is, this is the RNA and this is the FUMP. So the fungus thinks, okay, this is uracil, and it takes it in. But the fungus does not know that this is not uracil, this is something that mimics the uracil. So it mistakenly takes this stuff into the RNA. Now what happens in the RNA? As we all know, RNA is responsible for the production of proteins. Now if it was actually uracil, it would have produced proteins, but in this case it's not. It's just something that mimics. So what does it leads to? It leads to the production of faulty, oh my god, I can't even write faulty properly. So it leads to the production of faulty, I hope my spelling is correct. So it leads to the production of faulty, one second, oh my god, faulty and defective proteins. And the rest is pretty easy to understand. Once the fungus produces defective protein, it's pretty obvious that they cannot even function the basic process, and it will ultimately lead to cell death. So what happens when defective proteins are produced? It causes disruption of essential cellular process. So cause a disruption of essential cellular process. I mean, it's pretty obvious, without proteins are the basic building blocks. Without the basic proteins, you can't do anything. So it leads to the disruption of essential cellular process and cell death. Now to come to the first, now this is how it works. This is one pathway which it can take, RNA synthesis inhibition. Now coming to the DNA synthesis inhibition, that is the second pathway which it can choose. Now over here the drug gets metabolized into what? It gets metabolized into 5-FUMP as the previous one suggested. Now this is metabolized into another component. What does it get metabolized into? It gets metabolized into 5-fluorodeoxyuridine monophosphate. I know this word is very long. If you cannot remember this, just remember its abbreviated version, which is 5-FDUMP. I mean, it's kind of similar. It's just that fluorouridine triphosphate. Over here it's fluorodeoxyuridine monophosphate. Slight variation is there. Here it's 5-FUMP and over here it's 5-FDUMP. Now what happens here? It does not mimic it doesn't mimic or fold the fungal cell as such, but this molecule itself is an inhibitor of the enzyme thymidylate synthase. So this is probably a very vital enzyme. So this molecule, it just comes and inhibits this thymidylate synthase. Now what is the function of thymidylate synthase? Nothing much. This enzyme is required to produce what? To produce, I'll just write it as caps. It's required to produce thymidylate. Now since this enzyme is getting inhibited, you no longer produce thymidylate. Now you must be wondering, why do you need thymidylate? Now thymidylate is nothing but a it's a building block of DNA, or I would say it's a component of DNA. So again, impaired DNA synthesis. Now the rest is pretty obvious. Without DNA synthesis, you can no longer go and causes cell death. If the DNA itself is not produced, then how will the cell survive? So this is how basically this classification of drugs work. Two pathways it follows. One is by RNA inhibition and one is by DNA inhibition. So yeah, this is how it undergoes. You have to remember the key points. 5-fluorouracil is very important. Flucytosine is the key drug that comes under the class of what? Antimetabolite. Now quickly going to the final classification. I know this video has taken a lot of time. It's probably because it's my first video. Now coming to the second last classification. This is not over. We have one more left, but I'll make sure it gets over now. Now the second last one is known as azoles, as you can see. Now I have given another image for azoles over here. I may or may not use it. May or may not use this image. I'll try my best. Okay, now coming to its mechanism of action. I'm pretty sure you must be tired of seeing this, but I'll make it interesting. Now simply what I'll be showing right now is how ergosterol is produced. Now ergosterol is produced from various processes. You have a thing called squalene. Okay, now coming to the first step, you have squalene, and squalene gets converted or metabolized into squalene epoxide. By what? Squalene epoxide by using an enzyme known as squalene epoxidase. So this enzyme converts squalene into squalene epoxide. The cycle is not over yet. Now the squalene epoxide gets converted into another structure called as lanosterol. What I'm showing you right now is the synthesis or the cycle for the production of ergosterol, and then only I can show you how and where the drug acts. Now lanosterol is converted into our final product. Now what is the final product? The final product is ergosterol. Now this is a cycle, or the cycle how it acts. Now there's an enzyme over here, I forgot, and the enzyme which converts lanosterol to ergosterol is known as lanosterol 15D methylase. Just move it a bit side. So this is the enzyme which converts lanosterol into ergosterol. Now this is how the ergosterol is produced. Now you must be wondering, where does this drug act? It's simple. Now just look at where I'm going to drag this. Aha. So as you can see, a drug acts on this enzyme. So yes, so this enzyme gets inhibited by what is the drug? Azole. So the azoles mainly act on this enzyme. So once this enzyme is inhibited, you no longer can produce ergosterol. Instead, the cycle will end with lanosterol accumulation. Now what happens over here? It's pretty simple. Nothing much. Over here you can see, like I've given here, there is very less amount of ergosterol over here. See, as I've written here, decreased production of ergosterol. Why? Because again, as I've shown here, due to the drug, the class of drug, which is basically azole, it acts on this enzyme, and therefore the cell or the production cycle for the ergosterol stops over here. So ergosterol is not produced, blah blah blah. You come to the final step where you don't have much ergosterol over here, and what does it lead to? It leads to nothing but disruption of fungal membrane, fungal membrane structure and function. As I told you, ergosterol is a very vital component of the fungal cell membrane. Without it, it will lead to a lot of various osmotic instability, and ultimately it will lead to the cell death. So this is how this class of drug works, which is azoles. Now coming to the final one, I hope I've written something over here. Let me just check. Now this is the final one. I promise you this will be over right now. This is allylamine. This is how it works. Now I've written some points as I've always written: active against dermatophytes and candida. Okay, let me just check if I've written anything over here. Nope, nothing. Okay. I know this is a bit lagging, but stay with me. So this is allylamine, which is active against dermatophytes and candida. Now its mechanism of action. Now I know you're surprised again. Before it was some other structure. Now I've changed the structure. Now this is the structure which I'll use for allylamine. Example, terbinafine. So I'll just duplicate this. So as you can see, this is a basic structure. Now I know all of you must be bored, but trust me, this allylamine follows the very exact mechanism, almost exact mechanism as that of azoles. This is a slightly different variation. As I told you, the mechanism includes squalene. Squalenes gets converted. Squalenes get converted into squalene epoxide. Which gets converted into lanosterol, and lanosterol gets finally converted into ergosterol. Now in the previous one, this was where azoles functioned. So there's a slight variation. If azoles worked over here, where it inhibited the formation of ergosterol. So what happened? Lanosterol used to accumulate. Now coming to allylamine, it works, as I told you, it works almost exactly similar. It's just where it acts. Now I told you there was an enzyme over here known as squalene epoxide. This was the enzyme. So what happens? Like I said, I'm pretty sure all of you must have guessed what happens right now. Now this thing comes and it inhibits squalene epoxide, and therefore squalene epoxide is inhibited. So it actually inhibits the first step of the cycle itself. So squalene gets accumulated. You do not form squalene epoxide, nor lanosterol, nor ergosterol. So like I said, it works in a very similar manner. This is the only difference between allylamine and the other one. And no, not really. So ultimately what happens, same thing. So what happens is that, let me just enlarge it. Now what happens is that accumulation of squalene actually occurs. Accumulation of squalene. What happens when squalene gets accumulated? It leads to increased toxicity, as squalene is actually toxic for the cell. Increased toxicity and increased permeability. Now increased toxicity is caused due to squalene, and increased permeability is caused due to what? Due to the lack of ergosterol. As ergosterol is responsible for the cells functionality and fluidity, etc., like I've said before. Due to the lack of ergosterol and due to the accumulation of squalene, you get two of this, which ultimately leads to what? Cell death. It leads to cell death. Now this is the last classification. Now we have completed all the four classification, that is polyenes, antimetabolites, azoles, and allylamines. In this, allylamines and azoles follow a very, very similar path for their mechanism of action, whereas polyenes and antimetabolites have a comparatively different mechanism of action. So this marks the end of the video. As you have seen, I'll soon come up with another video covering more topics from antifungals. So yeah, thank you for watching.